Hello everyone, my name is Yaroslav Sadovский. I work at the Laser and Plasma Institute of the National Nuclear Research University MEPhI. At the Plasma Physics department, I give a lecture course named Methods of Surface Analysis. And by surface, in this context, we mean any kind of solid body or sample, and it's very essential for us to examine these samples before and after experiments, so we use different methods of examining the samples. There is a huge variety of methods of analysis of surfaces in terms of composition or surface shape or surface relief, and that's what we are interested in at our department, and any scientist working with plasma impact on the surface, coating, deposition, and so on. So a high variety of methods giving these possibilities exist. But one that we are going to talk about today is by far the most popular and gives the most straightforward results, because the results it gives us are not figures, not graphs, not plots, but images, actual images that we can look and examine. I'm almost sure that you have seen such images either in scientific papers or in the news or somewhere else on the internet, because they look like ordinary photos, grayscale photos, and they have all the distinct features of ordinary photography, because they are grayscale, they have shadows, highlights, and so on. They have even depth of field effects, blur backgrounds, and so on. So they look just exactly like ordinary photos in grayscale, but they are not, and they are not obtained by any means of optical imaging techniques. They are obtained by a device called electron microscope, and in particular Scanning Electron Microscope, or SEM. That's the theme of today's lecture, the Scanning Electron Microscope, SEM. I will try to give you basic information on how it works, what are the main principles of operation, and basic device construction and parameters, and so on. So what's the difference, and why cannot we obtain such images with such high magnification in using optics, using ordinary optical microscopes? The limit is quite simple, and it is dictated by physics. As you know, the wavelength of visible light is in the range of hundreds of nanometers, somewhere in the range of 400 to 500 something. So any object that is smaller than that would not be seen by optics. It would be actually transparent for any kind of light wave of that wavelength. So to be able to get an image of objects that are smaller than that, you must use something that has wavelengths smaller than that. That's quite obvious. And by the start of 20th century, it was already known that an electron behaves both like particle and wave at the same time. And since it behaves like wave, it should have some kind of wavelength, right? That wavelength depends on the energy of the electron. If we give an electron an energy of several hundreds of EVs, or larger than 1 keV, the wavelength of that electron would be much smaller, or in the order of nanometers. So in theory, we would be able to obtain images of objects of that order of size, nanometers. And that's what is used in electron microscope. Historically, the first electron microscope to be developed was transmission electron microscope, or TEM. The principle of the operation of TEM is quite simple, but we won't discuss that into details for now, because in my opinion, scanning electron microscopes are more widely used. They give more informative results in terms of images, and their images are easier to interpret. So scanning electron microscopes were developed several years after the development of TEM. That's in the range of 1930s to 1940s. And commercial SEMs were already sold in 1960s. And since then they are developed and upgraded and so on. Different add-ons are being added, characteristics are being upgraded, and so on. So modern SEMs are quite advanced devices which give opportunity of obtaining images of very high quality and very high resolution in the order of nanometers or even less. So the principle of operation of a scanning electron microscope is quite simple, and I will try to explain it in a very brief and basic terms right now using this pen and a notepad. Let's imagine that this blank notepad is a sample which we want to examine and get an image of, supposedly magnified image, because we want to achieve some kind of magnification. So this pen is an electron beam coming from the top. The third thing that we need is some kind of detector, because we want to detect outgoing signals from the sample, which in this case would be secondary electrons. Secondary electrons are electrons that were knocked off their positions from the sample. So they are electrons belonging to the sample, which were knocked off by the primary beam. And we will detect them. The primary beam usually has energies in the order of several kiloelectron-volts. Secondary electrons, electrons which belonged to the sample and were knocked off by the primary beam, usually have energies in the order of several EVs, 5 to 10, maybe 15 EVs. So to catch them, to gather them, we would place some kind of detector to the side of the sample. And we also would place a conductive grid made of wires in front of that detector. And we would bias that grid so that electrons going out of the sample would be drawn towards that grid. So in this case we would collect almost all secondary electrons, but that's the case here, the word almost. So if the sample is quite plain and uniform and homogenic, no matter what spot on the sample surface we hit with that primary beam, we would collect secondary electrons in the detector. And no matter of the spot, the number of secondary electrons gathered by the detector would be almost the same. So the signal would be almost the same in each and every case, and we won't get any kind of image. But let's imagine that the sample is not uniform. Let's imagine that it has a very sharp a sharp ridge on the sample, on the surface. In this case, the situation changes. When we hit the side of the ridge that is facing towards the detector, the situation is quite the same. Almost all secondary electrons emitted from the surface would be gathered by the detector, and we would obtain a high signal. If we hit the side of the sample, the ridge that is facing not towards the detector but to the other side, secondary electrons would not sense the field of the grid and would not be drawn to the detector. So they would, most part of these secondary electrons would escape and go towards the walls of the vacuum chamber. I didn't say that, but obviously the whole experiment is conducted in a vacuum, because you have electron beams and free electrons with quite low energies, so you need good vacuum conditions in order to do that. So the difference is in the signal that we are registering on the detector. In this case the signal is high, in this case the signal is low. And when we start moving this probing electron beam, the primary electron beam, across the surface line by line, that is called line scanning, hence the name of the method, scanning electron microscope. We scan the sample with an electron beam, and at each spot we are measuring the signal that we are gathering on the detector. And simultaneously we are coloring pixels on our virtual image of the sample. So let's say the starting point is top left corner of the sample. We are obtaining quite low signal there, and we are coloring the pixel on the image respectively in any kind of palette, but usually grayscale palette is used in secondary electron microscopy. So if the signal is low, we color the pixel black. The situation is almost the same as we go up this ridge, but when we reach the top and go to this side, the signal intensity changes because almost all secondary electrons now can reach the detector because they are all going in that direction. The detector is here, we catch almost all the secondary electrons and we obtain quite a high signal there. So when we start coloring the pixels on the top line, on the right part of that top line, we are obtaining high signals here, and we are coloring the pixels white or light gray, if you will. And then we do the same for all the lines. And we end up with some kind of image that consists of pixels colored in all shades of gray. The question is, will this image reflect in some way the surface relief of the sample? And I'm almost sure that you already have the answer. Yes, it will, because it looks like a ridge. This image would look like a ridge. The left part would be black or gray, the right part would be white or light gray. And it would resemble the object that we were scanning. So by the end of the scanning process, we end up with an image that surely resembles the sample surface. But what about magnification, right? So the whole idea of the scanning electron microscope is that we get a magnified image of the sample. Well, magnification is defined as a ratio of sizes of size of the image we obtained on the display and size of the sample that we were scanning, the scanning area that we were scanning. In this case, if we had the display of that size and the scanning area was almost the same size, we get no magnification. The magnification factor was 1x. If we wish to obtain a magnified image of some area of the sample, the only thing we need to change is the size of the scanning area. We won't be scanning the whole sample, but we will be scanning a very small region on its surface. The most interesting region is the top of the ridge here. So if we wish to have a magnified image of this top of the ridge, we would be scanning maybe a square centimeter size area here. And we do all the same things, measuring the signal intensity and coloring the pixels respectively. So when we are finished scanning the square centimeter region here, we end up with an image on the display of the same size and same amount of pixels. And in this case, the image would be magnified because the size of the image on the display is much larger than the size of the area that we were scanning. In this case, I assume that would be like a factor of 10 or 15, which is not that high. But the nicest thing about scanning electron microscope is that we can achieve quite higher magnification factors in the order of tens of thousands. And the simple answer to that is just reducing the scanning area. Let's suppose we were scanning an area 5 microns at each side, 5 microns square. We would move the electron beam in very tiny increments, let's say maybe 10 nanometer per pixel. So we measure the intensity, move the beam slightly by 10 nanometers, measure the signal once again, and so on. The same process, and we would obtain an image. The quality of that image is the question, and it highly depends on various parameters of the beam, and we will talk about that slightly later. But the magnification would be quite higher. And that's the idea of scanning electron microscopes. That's how we obtain images. The magnification only depends on the movements of the beam, the size of the scanning area. I will now try to show you the basic scheme of the apparatus itself, the basic components of the microscope itself. So let's draw them here. To start an electron beam, we need an electron gun. The main element of the electron gun is a cathode. We will talk about cathodes on the next slide, but as for now, the cathode is the start. Then we need some kind of preliminary accelerating system for the beam. That's where the beam starts. Then we need some kind of focusing system and apertures in order to form the beam. In actual microscope there are a lot of lenses and apertures there, but I will draw quite simple here. The almost the final stage are some coils, magnetic coils, that actually shift the beam, the coils that deflect the beam and enable that scanning motion. The final stage is the sample stage, the table on which the sample sits. Usually the table has controls of movements in 2 or 3 axes and rotation maybe in some devices. And the whole thing is inside a vacuum chamber. So the whole thing is inside vacuum. Sometimes there is a lock chamber here to enable quick sample exchange. But the basic scheme is such: an electron gun, beam preparation system, beam deflection system, sample stage. The final element is the detector, or detectors. There could be multiple detectors here, which detect secondary electrons emitted from the sample. The only thing that's left are control electronics and imaging electronics that enable gathering of an image and drawing that image on the display. So for cathode choice, there are usually 3 main options. The first one, the most simple one, is a heated tungsten wire cathode, which is just a tungsten wire shaped in a V shape, which is heated by current passing through it, heating it up to quite high temperatures so that it glows white color, and thermoelectronic emission occurs, and we could then draw these electrons off the cathode and direct them and form the beam. This type of cathode is the least expensive one, the most simple one, but it gives not the best quality beam, not the best shape of the beam, not the best control of the beam, and so on. So the next option is a lanthanum hexaboride crystal, which is also emitting electrons when heated, but to less temperatures, and the shape of the crystal could be controlled better than the shape of the bent tungsten wire. So the quality of the beam is better in this case, but the bad thing about lanthanum hexaboride is that it reacts with oxygen, and vacuum control must be quite strict in this case. The final option is a so-called field emission cathode, which is just a needle, but a very thin one, which emits by means of auto emission in quite a high electric field intensity. If we apply quite high electric field intensity to a very sharp needle, the electrons would start to fly off the tip of the needle, and that's it. No heating, auxiliary heating required in this case. This type of cathode gives the most thin and narrow beam, but it has its own drawbacks also. Not the best intensity could be drawn out of this type of cathode. Now I have a question for you regarding the interpretation of the images obtained on the Scanning Electron Microscope. Since now we know the basic principle of operation, we know that there will be light and dark areas on images that we can call shades. The dark areas could be called shades, and shading occurs naturally. The image looks like if it's irradiated by a light source, but there is no light source obviously, but we could imagine that there is a virtual light source somewhere else. Can you guess the placement of that virtual light source? The correct answer is that the virtual light source is the detector, because regions that are not facing the detector would look dark on the image and thus cast shades there. So the virtual light source is the detector. The camera, the virtual camera, the point from which the image is obtained, the point from which we are looking on the sample, is the position of an electron beam. So we are looking from the position of an electron beam. And this may give some confusion when looking at some images. Take a look at this one and try to guess if it's a pyramid or the opposite. When not knowing the placement of the detector relative to the sample, you can make an error in this choice. But when you know the geometry of your experiment, then you may be sure about the answer to that question. Now we will talk about basic parameters of the beam and scanning system and their effect on the image quality. Among these, the most core ones are beam energy, that is the energy of the electrons in the primary beam, beam current, which is the amount of these electrons in the beam, the intensity of the beam, the current of the beam, and scanning speed. These are three main parameters which have very drastic effect on the image quality, and their combination is essential for a good quality image. So beam energy. One might think that the higher the energy, the better, and that's partially true. The higher the energy, the easier the beam is to be moved. It doesn't bend, it moves in a straight line, and it's easier to pass through the whole system through bad vacuum conditions and so on. But the size of the area from which the secondary electrons are being emitted is growing as the energy of the primary beam increases. And this may result in very interesting image quality changes. In this example, you can see one and the same object being captured with different beam energies. You can see that at high beam energy the object looks like, if it's semi-transparent, it looks like some kind of gel, but it's solid. And that's the effect of secondary electrons being emitted from the volume of the sample. And since they are emitted from the volume and we're detecting them by our detector at each and every pixel, the object looks like a semi-transparent object. It looks like gel or something like that. Sometimes this effect is desirable, sometimes not. If you are caring about very small details on the surface that may look transparent to the beam, you must lower your energy so that these details does not look transparent. But lowering the energy is quite difficult because a beam with lower energy is harder to steer, harder to focus, and harder to manipulate. So you have a choice. They're either low energy but quite hard maneuvering with your beam, or high energy but maybe some loss in very fine details of the image because they become transparent. Usually the range of energies used in SEM is from several keV, 1 to 5 to tens of keVs, 15, 20, 30 and so on. And the choice of the energy depends on the sample, strictly on the sample material, on the size of objects and so on. The second main parameter is the beam intensity, the beam current, that is the number of electrons in the primary beam. Once again, there is a compromise here. One might think that the higher the intensity, the better, and it's also partially true. Of course, you would gain in intensity. Your signals in any range would be higher and easier for detection and amplification and so on, but a beam of high intensity cannot be very narrow because electrons start to repel each other and the beam cannot be focused to a very tiny dot on this sample surface, and thus it would also affect the image quality. We would get blurry edges and low focus and so on, and we won't be able to get very high magnification because of a low resolution. The spot of the image defines the resolution. The spot size on the sample defines our resolution. If the beam is focused to a dot of, say, 3 nanometers, that's the best resolution we could get. Because in other case, you move the beam less than the beam diameter, you won't get another signal. You won't get a different signal. You will get almost the same signal from a sample surface, and that would result in blurry edges of the image. So once again, there is a compromise here. You may choose the low intensity beam for high resolution, but thus your signals would be lower, harder to amplify, a lot of noises and so on. But you may choose high intensity beam and then get good quality signals, high intensity signals, but lower resolution. That's the second compromise here. The third main parameter is the scanning speed, which is the rate at which the beam scans the sample surface. This could be also varied, and the effect on the image quality is noise reduction. Because the more time we spend on each pixel gathering signals, gathering secondary electrons, the more statistics we get and the less noisy the signal is. So if we spend, let's say, 1 second at each pixel, we won't get any noise because statistics would be very high. We would get high intensity signals, high statistics per each pixel. We won't get any noise, but the gathering of the image would be quite a long process because you're spending 1 second per each pixel, and there is, let's say, 1 million pixels in a good image, so that's 1 million seconds. So there is a compromise here also, the third one. If you desire to get your image quicker, you induce quite high scanning speed, and that means lower statistics per pixel and possibly noisy image. It would look noisy, not very uniform colors and so on. Just like in photography, by increasing your ISO sensitivity, you would get a good image in dark conditions, but it may be noisy. The same thing goes here. So there are three compromises here: beam energy, beam current, and scanning speed. Is there a universal recipe for a good quality image using SEM? Well, yes and no. There are some conceptions of obtaining better quality image, but each case is specific because it is defined strictly by the sample, by sample materials, by the desired magnification rates, and so on. I will try to tell you the best recipe scenario right now. So in order to obtain a good quality image in high magnification, the basic recipe is such. You need to have quite low beam intensity in order to obtain a very narrow and sharp beam. You need to have quite long scanning time or low scanning speed in order to obtain good statistics. And you need to tweak your beam energy in such a way that objects that you are analyzing do not look transparent or semi-transparent. And that's the basic recipe, and all the other parameters could be varied, but the main thing is as such. And I think we may conclude with that. So that's the basic information about scanning electron microscopy. The basic principle of operation, basic scheme of the device, basic parameters, and their effect on image quality. I hope you all now understand how these images that you most surely have looked upon in scientific papers, how they are obtained and were obtained, and how to obtain a good image if you are working on this device yourself. So I hope you learned something new today in this lection, and you will carry this on, and please try learning something new at this time when you spend so much time home. And I hope you are all healthy. Take care and goodbye.